observing special gene expression patterns in the developing mouse brain using house trees. House trees. House tree stands for Hierarchical Orientation Structural Tree. Leverage the Allen Developing Mouse Brain Atlas, used in a tool for interactive exploration of special gene expression and overall structural developmental patterns, will enable users to answer all questions posed by 2013 Scientific Visualization Contest. Allen Developing Mouse Brain Atlas, a recap, was developed to explore how gene expression changes throughout the process of development in mouse brain. Has about 2,000 genes functionally relevant to brain development or disorders of the brain. Provides ISH energy data across 3 prenatal and 3 early postnatal stages. Provides age-matched anatomic reference atlases. Contest questions. According to the contest description, 6 volumes of structure annotation IDs are given. Each volume provides structure IDs for each stage, including 3 prenatal and 3 early postnatal stages. At the same time, gene expression energy are also collected. Therefore, based on the structural patterns and the gene expression patterns in development, how to answer the following questions: gradient identification, structural patterns, structural consistency, and complementary patterns. Data collection. Through the data collected, we are focusing on the following content: stages, annotation volumes, structure levels, structure IDs, gene categories, expression energy, and genes IDs. Working with data. To achieve our goal to answer all contest questions, we first build our visual analytic system to implement a one-time data processing, and then use visualization tool to present the patterns and hence to answer the questions. Visual analysis. The goal of visual analysis is to filter the entire content, to find the correlated gene expression structure sections, and to make the data efficient for visualization. Gene expression flow. Through the data, for each gene in each structure, a time-varying expression signal could be found. Next, we use Pearson correlation coefficient to compare this flow with 5 typical shapes, try to find the most similar one, and use the color to label it. Taking gene chat in structure ISP as an example, expression signals made such a flow correlated to the last flow curve. So, chat in structure ISP is labeled as blue. Gene expression structure matrix. Totally, we have 2,105 genes in 2,692 structures across in 6 stages. Gene expression flow matrix. A flow-shape-based 2D matrix is shown here. Genes are in rows, while structures are in columns. Each element is colored by the flow shape label. Biclustered gene expression flow matrix. In order to find the potential gene structure patterns, an Euclidean distance-based biclustering method is used to search for the correlated sections in the expression flow matrix. And now, the data are efficient enough for visualization. Visualization. The purpose of visualization is to explore spatial gene expression and overall structural development patterns interactively, and to answer all questions. House tree. The house tree is constructed based on the hierarchical development levels. We use node to represent each structure, and the edge connect between two nodes to indicate their hierarchical neighbors. The whole tree are constructed in concentric rings pattern, and the hierarchical levels are used to serve as the corresponding radius. Color in the house tree. Inspired from diffusion tensor imaging, we use a color system to indicate the potential development orientation to find the spatial patterns. For each available structure volume in annotation, the center is calculated. In this case, the vector from preceding to succeeding structure centers denote the potential development orientation. The 3D vector is converted into RGB color by normalizing X, Y, Z into red, green, and blue. In addition, for those interactive exploration. Now, we are going to use house tree to explore spatial gene expression and overall structural developmental patterns. First, we are focusing on a section has consistent expression flow. The corresponding genes and structures are shown in the matrix. At the same time, on the right, the corresponding structures are highlighted with the circle. We found the structure mostly located in an extremely wide range between 5th and 10th level, and the edges among them are in various colors. Since the neural system has the longest developing period, as well as the diffuse orientation, we hypothesized the correlated genes potentially related to neural system development. To prove our hypothesis, a category histogram of these genes is generated. It clearly shows that this gene significantly belongs to transcription factors, nervous system development, and neurological system development, which proved our hypothesis. Next, we choose another section displays an intensive blue color. From the highlighted structure node in the tree, we found these structures mostly distribute in a relatively late range, between 9th and 10th level. We believe these correlated structures are primary regions where synapses developing in this range of stages. Answer the question. Gradient identification. House tree use the structural development directions to present the spatial expression patterns of the gene correlated to those structures. Structure patterns and consistency. The biclustered matrix uses a color label to indicate the expression flow classification over stages in any given structure subset. Thus, one can observe structural patterns as well as consistency through the time-varying color in the matrix and the spatial orientation color in the house tree. Complementary patterns. For each given structure, the corresponding structure column of the biclustered matrix present complementary expression patterns in the same color label. Conclusion. In conclusion, house tree is used in a tool for interactive exploration of spatial gene expression and overall structural developmental patterns, and it answered all questions posed by 2013 Science contest.